Okay. So I'll end the poll. We'll start up today's class. 1 second, give me a second. Yeah. Okay, people. So today we have our topic as transgenic animals. So we are talking about biotechnology and its application. So this is a very important topic when it comes to development of products based on the application of biotechnology. So to understand this topic, initially, we'll have to check our basic knowledge. So I will ask a question for you. So you can answer this question in 1 minute. Okay. So abnormal gene is replaced by a normal genes through gene therapy, medications, cloning, and radiation. So you have 4 options. So I'll give you a minute to answer this question. So let me launch an MCQ poll. So you have 4 options, right? So 1 minute, you try to answer this question. So once again, give me a second. Yeah. Yeah. So when an abnormal gene is replaced by a normal genes, okay, what method do we use? So you have just 20 seconds. Okay, students. So I'm going to end the poll. So you have your answers anyways. So let's check what's the right answer for now. Okay. So one of the major applications of genetic engineering and biotechnology today is, and it's also a cutting edge technology because when we see most of the congenital diseases like Alzheimer's, okay, rheumatoid arthritis, or you have cystic fibrosis or phenylketonuria, sickle cell anemia. So there are so many genetic disorders which cannot be cured by normal medication. Why it is permanently not cured is because it is a effect or there is a problem with the gene itself. So whenever there is a problem at genetic level or gene level, so it is hard for any of the solutions which can be brought for a permanent change. Okay, so medications are some remedies that can help us prolong the life, and it does not cure the particular disorder completely. So it doesn't happen with medications, right? 1 second. So medications, it's not possible. So when it comes to cloning, so what is cloning? Whenever we have a recombinant DNA technology on a particular cell or a particular tissue or a particular organism, let us say, or if we have a hybrid, okay? So whenever we have a particular genotype and we try to preserve that genotype and we create an identical offsprings, which are genotypically and phenotypically very similar. So this is called as cloning, okay? So cloning is one technique where we try to preserve the genes of a particular organism. Okay, so the cloning is not possible, right? This is not talking about curing or replacing an abnormal gene with a normal gene. And the final option we have is radiation. So when it comes to radiation, radiation is one of the types of therapies we use to alter, okay, or kill certain cells whenever they are not having a normal genes. For example, cancer cells, right? So we actually apply radiations to these cancer cells and try to eliminate them from our systems and without affecting our normal cells. So that is radiation. So here we get rid of the cells. We don't actually correct those cells. So radiation is not the right option here. So when it comes to gene therapy, as I said, it is one of the most cutting edge technology when it comes to biology, and especially when it comes to molecular biology. So here we use several techniques like vectors and recombinant DNA technology, right? And we try to preserve the normality of the genes by replacing these abnormal genes from those cells. So this entire process is called as gene therapy, okay? So let us see the second question we have here. The first human hormone produced by recombinant DNA technology is insulin, estrogen, thyroxine and progesterone. So you have your options. So let me launch a poll for you, right? Give me a second, people. Yeah. Let me launch an MCQ poll. So you have 1 minute to answer this question. It's already, if you're wrong, just try to answer so that you can memorize very easily. When you participate, it becomes very easy for you to memorize, okay? So which is the first human hormone produced by recombinant DNA technology? Nice. So you have your answer anyways. So let's see. So some of the questions that comes in the examination are factual based, and this cannot be changed. It has to be based on your practice and memory. So your answer was right, okay? The first hormone that was produced by recombinant DNA technology is insulin. Okay, it's very popular because we also have a topic on how to produce a recombinant DNA technology, insulin, right? So anyways, so let's get to the topic, people. Today, as I said, we are talking about transgenic animals, so which is very important for us today to actually recognize this particular topic. Okay, so we understand what is biotechnology and how do we develop recombinant cells and how do we create the products from the recombinant DNA technology. But one of the major parts that we are missing is after when we talk about recombinant DNA technology and its application in human welfare. So this topic is very sensitive when it comes to human applications. So for example, if we have to study any of the characteristics of a gene with respect to its physiological expression, okay, or whether it comes to the disease, like or how the disease manifests, or how do we identify a disease, okay, or to study the characteristics of a disease, or for example, you can talk about some of the diseases which are like Alzheimer's, or you can talk about congenital disease, any of the congenital diseases you take. So it becomes very hard for us to look only at the physiological perspective, but when we have to know the complete details of the disease, like how it is manifesting and what are the factors that is affecting to cause a particular condition like this in humans. So we have to take a deeper look at it, okay? And imagine that we use human models to study these particular aspects. So it's very, very impossible to study diseases like this or certain conditions or certain production of certain products on a human scale. Not only on human scale, since it's very sensitive when it comes to the biology of a particular, when it comes to biology as a whole of a biology, it's very sensitive topic. Why? Because science is different and ethics is another thing that binds the science with what we do with biology, right? Correct, no? So because we have the capacity to understand something and we have the capacity to influence something on organisms, that doesn't mean that we have the freedom to do it on all the organisms that we have around us. So based on ethics and based on the technology that we can implement, so we create a new branch where it becomes helpful as well as it is ethical for us, and that is where the transgenic animals comes. So these are our beneficiaries, and we have to thank them for their sacrifice. So that because of the transgenic animals only today we are able to study so many aspects of genetics when it comes to humans and produce products, okay, biologically important products, understand diseases, okay, and so many different applications that we have developed based on the use of transgenic animals. So some of the common transgenic animals that we use to understand this is, for example, rats or mice. So the rats and mice or mice, okay, they are the largest transgenic animals, okay, that are being used for this particular purpose. So next one is rabbits, pigs, sheep, cow, and fish. So these organisms are mostly used as transgenic animals. So when you look at the the models, like transgenic animals are genetic models, isn't it? So when you look at these organisms that we use, okay, to test certain things or to understand certain things, okay, so this is based on the overall acceptancy, okay, the population, the entire human population and their consciousness for life, or overall concept of how do you preserve life and how do you respect life. So these organisms are chosen because it is quite acceptable to a certain level. Okay, so I'm not telling that 100% of the people are okay with it. So instead of going to the organisms that are rare, exotic, and very hard to work on, so these organisms are chosen on various aspects, maybe because of their size, maybe because of their ability to reproduce and create more and more offsprings. Okay, so something like this. Okay, so as I said, 95% of the animals that are used as transgenics are mice. Okay, so one of the reasons why we use these particular models or only these specific organisms is because of the easiness to raise them. Okay, one of the major things. And the second thing is the number of offsprings, the number of variants, their small gene size, and all the aspects come into play when we talk about transgenic animals. So why do we use transgenic animals basically? So we have 5 applications, broad applications we can tell when it comes to the when it comes to the use of transgenic animals. So let us see the first one. The first one is to study regulation of genes, okay, which is a very important aspect for us, correct? So when we talk about regulation of genes on normal physiology and development, what it is very curious for us to understand, okay, human beings have such a vast amount of genetic information. For example, when you talk about DNA, you can see the volume of DNA is very, very large. But when it comes to the expression of DNA, we have a significantly a quite large number of genes. For example, 25 to 30,000 genes we have. So when we have that amount of genes, it is very essential for us to understand how these genes behave and what are they contributing. What is their contribution in the expression? What contribution do they bring in the expression when it comes to us? Not only in us, with respect to other organisms and other genetics also. So you just imagine like using the same models on humans. Okay, so it is very hard for us to understand the entire physiology and keep on straight, because it takes a very long time, and you cannot use the subject for a very long time. You can't make a person to stay in a lab for so many years and study their physiology, right? So that is why we can take our genes and put it in the organism, and we can study the physiology and how it influences our development. So that is the first thing. And second thing is how the genes contribute in development of a particular disease. So most probably we use this application when it comes to congenital diseases. So congenital diseases, their characteristics have to be studied at physiological levels. And since they are not pathogen based and they're not infectious, so it is developed in an individual because of a failure of certain physiological characteristics, which are again related to genetics. So it is very essential for us to study these diseases so that it's very easy for us to develop cure or remedies to cure, remedies for these particular disorders. Next one is the very important aspect is development of biological products. So today, millions of lives across the globe have been saved because of the development of biological products. On various levels, we use the biological products to, in human application whenever it comes to medicine. It might be enzymes, it might be hormones or a particular proteins, okay, or particular supplements or certain factors that are responsible to accomplish a biological process. So that is where it comes, okay, development of biological products. Next one is vaccine safety testing. So another problem that we face today is the disease, right? So the diseases spread and they can create a very tremendous amount of problem. If you take the recent examples like COVID-19, so you understood the rush for developing vaccines. So developing vaccines itself is a very sensitive process. And after developing, you cannot just think that these vaccines can be directly put for human applications, because we are using the live organisms or a part of these organisms which can be very sensitive to our immune system. So that is why the testing and thorough processing is very essential to make sure that it is very safe and it works on human subjects, right? And finally, we have is chemical safety testing and toxicity testing. Whether it might be particular, whether it might be particular drugs, or it might be cosmetics. Okay, it might be anything. So when it comes to human application, again, as I said, it's not possible because it might lead to severe controversies and also certain irreversible problems in our society or population. So that is why we test on certain model organisms and we make sure it is safe enough for the human use too. Okay, so let us see these applications in quite detail. So when it comes to regulation of normal physiology, as I said, okay, for example, if you take any hormones or proteins or enzymes, so they might influence on several levels. For example, your growth factors, okay, human growth hormones or insulin like growth factors. So their complexity is way too high to understand on, it requires an elaborate investigation onto particular influence of particular of these products, I can tell. Okay, so what do we do is we take the genes of that are responsible for producing certain hormones or enzymes, and we put it in the transgenic animals. As we introduce into these organisms, so we can understand how these genes manifest biological changes, and we can study these subjects. Okay, so this information is very vital for us to understand how our biology works and how we can use this, okay, knowledge to correct in abnormal cases. Okay, and next one is when it comes to the development of, give me a second, development of a disease. As I said, genetic models are key for us to understand many of the congenital diseases. So one of the biggest challenges that we have is cancer. So how do we understand cancer? So regularly taking the human cells or cancer cells is very hard, and we can manifest cancer, or we can actually induce cancer-like conditions onto these subjects or transgenic animals. For example, how do cancer cause, or like why do people get cancer? Is there a particular food or a particular product in the food, or it might be certain carcinogens, okay, or it might be particular medications. See, even though we use medicines for curing diseases, certain drugs or certain medicines can induce diseases, right? It is possible, and it can induce cancer too. So now we can use this models to understand whether these conditions can cause cancer. What is the behavior of cancer cells? How do cancer spread, right? So we can use several dyes and staining methods, as well as identification processes to trace these diseases. Same goes with cystic fibrosis or rheumatoid arthritis or Alzheimer's. So in any of these cases, if we try to, if we have to understand, transgenic animals will give us upper hand in giving us an room to study them in very detailed, in a very detailed way. Next one is development of biological products. Okay, so biological products are life-saving, as I said. Take for example, India or Asian countries. So if you see the number of patients, diabetic patients in South Asian countries, it's quite high. Why? Because of our rice-based diet. So now, when we have a lot of diabetic patients, one of the most important products that saves lives for is insulin. So look at how many patients regularly require insulin on a daily basis, okay, to keep their physiology normal. So that means that it is saving lives on numbers of millions, like we can tell. So development of these products actually requires a lot of understanding about a particular hormone or a particular enzyme or etc. For example, take for example antitrypsin. So antitrypsin is developed to treat emphysema. Emphysema is difficulty in breathing. For example, we can tell, how do we understand emphysema? The people who smoke a lot, for example, smoker's lung. So they have difficulty in inhaling, basically fix the amount of oxygen that is required. That is called as emphysema. So using alpha-1 antitrypsin can cure emphysema. Okay. So we understand the disease phenylketonuria, what happens in phenylketonuria, because of absence of a particular enzyme that helps in converting phenyl tyrosine into phenylalanine. So that is why ketonuria occurs, isn't it? So now we can actually supplement these patients with the enzyme that is responsible for converting this. So then they can get a temporary relief, and also with respect to cystic fibrosis. Okay. So a lot of these diseases require biologically important products. Okay. And these products are developed from transgenic animals only. Okay. Next. So we have a history here. In 1997, you have to remember this. Okay. Definitely. 1997, Rosie was the first transgenic cow. Okay. So this cow was genetically modified to produce a particular type of milk, which is very rich in protein. So we call it as, we have a subject actually, when it comes to this, we call it as biofortification. So basically what we are doing is we are using this technology, okay, genetic engineering, to develop certain seeds and certain animals, okay, which are very rich in proteins and which are very rich in minerals, right? So that even if you eat a little, your body is not malnourished with any of these supplements that is required. All right. So this transgenic cow, Rosie, could produce the protein-enriched milk, which for every liter, 2.4 gram of protein could be supplemented for a human being. So this is very nice. Why it is very nice? Because for some people who are very poor, who cannot afford food, right, on a regular basis, who are having only few meals a day. So through government programs, whenever the milk is given, the enriched milk is given to the kids. So they will become stronger. They will become disease-free. They will not be malnourished. Okay. So it's very nice application. And one more application you see. So mother's milk is very important for immunity. And the second point is it's very important for nourishing babies. If a mother is very malnourished and she is not being fed properly, and she is not nourished properly, then the development of baby is also poor. Why? Because she cannot produce quality milk. And usually what happens whenever it is switched between mother's milk and cow's milk, the tummy of the baby hurts a lot because of a lot of lactose, right? So that is why the cows are designed, okay, they're actually engineered to produce a specific type of milk that is rich in alpha-lactalbumin. So this alpha-lactalbumin is very good for your babies. Okay, it will not hurt their tummies, and it will also nourish the babies, and their health will be very, very normal without affecting their gastrointestinal tract. So that is good application, isn't it? So these are all biological products you can see here, right? Whether you enrich proteins through milk or enrich alpha-lactalbumin, you enrich your crops, enrich the meat, okay, or produce hormones, enzymes, or whatever it is, it's all biological products. Understood? Next one is vaccine safety testing. So as I said, vaccines are very, very, very important. So why it is very important? Recent cases are the very good examples for us. COVID-19 is good example. Polio is very, very good example. Diphtheria, cholera. Okay, these are all life-threatening diseases. So before even we see, for example, creating antibiotics for this is very, very hard, and it is impossible. Let us take it. We don't have antibiotics for that. How do we cure a disease? Our body itself has a mechanism to cure a disease. How? We have to make sure that our body is aware of what disease it is acquiring, so that it knows how to fight the disease. That is the vaccine, right? So when we introduce the antigen which causes a disease in its inactive form, whether it is a whole organism itself, dead or alive, but the only thing is it cannot cause a disease, or it can be a polysaccharide, it can be a protein, anything is introduced into the host. Okay. So when it is introduced into the host, take for example polio is live attenuated vaccine. That means the organism is alive. Attenuation means it does not have the capacity to cause polio. Okay, live organism is introduced into your body. When a live organism is introduced into your body, imagine a vaccine safety testing fails, or it is not tested. If an organism is not attenuated properly, and then it is given to people, healthy people will become polio patient now. Correct? No. That is why vaccine safety testing is very, very important, and transgenic animals are specifically used for vaccine safety testing. Understood? And moreover, whenever we design drugs, or whenever we design, okay, it's coming here, chemical safety testing, or whenever we design drugs, or whenever we produce biologically important products, okay, so it is first tested on animals. Why? Because Take for example tuberculosis. Okay, the medication for tuberculosis is quite it is quite toxic for liver. So now what we do, we cannot directly give it to human subjects and we will tell, okay, take it for these many months, certain dosage. And after that, what happens? Our body has to get rid of these drugs on a regular basis or medicines on a regular basis. If the medicine is accumulating in our body, it can lead to severe toxicity and it can lead to kidney failure, liver failure, and it can affect our vital organs and normal physiology. So that is why what they will do is, so they will test on these organisms and they will tell, no, paracetamol, take 650 mg. How do you know that it is 650 mg only? It has to be taken. So that is called as knowing the levels of toxicity, knowing the dosage and everything. All that knowledge comes from transgenic animals. Okay. So we test the chemicals, we test the biological products or drugs on these animals to understand how much exposure is safe for human beings. Understood? So it can be cosmetics. Okay. A lot of people use cosmetics, right? So whenever imagine you use shampoo and you have severe hair fall or you have rashes on your skin when you use soap. So it doesn't happen. How do they know? How do they create such a balanced product? On a regular basis, they're tested on these animals as they develop this product, and that will alter the results for our safety. Understood? So these are the applications of transgenic animals. So now we have a question. Maximum number, okay, of existing transgenic animals is of, okay. So I will launch a poll. So you take a minute and try to answer this question. Students, I hope you're understanding. If you have any doubts, you can just ask me. It's quite easy, no? Topics are quite easy. So come on, we have only 20 seconds. Question is, maximum number of existing transgenic animals is of. So which is the animal which is used as a transgenic one in highest number? Okay, we have 3 seconds and I will end the poll now. Okay, so your minute is over. If you have anything or if you think that this option is right or wrong, don't mind. Try to answer it first. Okay, you don't lose marks here, you lose in the exam, no? Anyways, as I said, 95% of the transgenic animals that we use is coming from mice. So our option is B. Okay, so this question was asked in 2011 NEET. All right, so our next topic is ethical issues. Okay, ethics are very, very important in science, correct? Why? Because we are not alone. We share this planet with many living organisms. It is a very, very sensitive balance between all of us, correct? No, ecology is very, very important. Ecology helps us to survive as one family. So for millions and millions of years, this sensitive fabric of genetics has been evolved on this planet. Now, we cannot play God just because we have the capacity to alter these characteristics. It is very sensitive, isn't it? How many millions of years of modification it could have taken to get one particular character or a trait? You tell me. So now, animals knows and nature knows its own pace of altering this and creating certain characters or creating anything. So now when we have the capacity to do it in very short time, for one gene to change, it will take several million years, several thousands of years. Now, what are we doing? We are altering it in a very shortcut manner. So when we do this, it will create unpredictable change because our understanding of world is very this much, very, very small. And life is a very elaborate thing. So a small changes that we do in an environment can be a disaster. Okay, forget about genetic engineering and how it has influenced. Humans intervening in a particular ecosystem, like for example, just introducing water hyacinths because of their beautiful leaves and flowers, has resulted in, when you go to West Bengal and Assam region, you just see these plants everywhere in freshwater system. correct? No. So that is human interference, introducing one variety of fish into a pond. Okay, it can be an invasive species. It can completely eliminate or completely dominate the naturally occurring organisms and completely destroy the ecosystem. It is not one organism that is surviving independently. Each organism is connected to so many different animals around it, like bees, insects, ants, rodents, etc. Correct? So whenever we introduce genetically modified organisms into our environment or ecosystem, it can really destroy a very fabric of it. And that is why, if you see in India, we don't have genetically modified organisms when it comes to fruits and vegetables. We don't use them. Only cotton we are using, and only some plants we are using. Apart from that, when it comes to normal consumption, we have you seen any genetically modified organisms growing wildly in some forest? No, it does not happen. Why? We don't know what is the capacity of this organism, how invasive it is, how can it become a part of ecosystem, correct? So that is why many government organizations across the globe, for every single country, there are specific bodies which monitor this. Okay, so one of our body is our own government. We have is Indian government have GEAS. What is GEAC? Sorry, what is GEAC? Genetic Engineering Approval Committee. So whenever you want to perform a genetic engineering experiment, or if you want to develop, if you want to develop a product, it is not possible for you to independently start your experiment. Even though it is easy, even though now the tools have become very simple and simplified and fast, you can't do it until there is proper approval, proper guidelines to what organism you can do, to what extent you can engineer a product, okay, or engineer an organism. All that is monitored by these committees. Okay, so now their decisions are very strictly based on genetically modified research. They prevent it, okay, from misuse of it. Okay, so these genetically modified organisms are quite dangerous. Okay, for example, you might end up creating a bioweapon, or you might end up creating some plants or some fishes or some microorganisms that can be unstoppable in our natural environment. So that is why these bodies will prevent from unethical use of these genetical engineering tools for their own selfish needs. Okay, next one is biopiracy. Now what happens is, biopiracy means, now whenever we have naturally occurring resources in our biology or with respect to cultures, okay, there are lots of tribes and there are a lot of indigenous people who have very exceptional knowledge about their ecosystems. Then they understand Ayurveda, okay, the knowledge about plants, their medical properties, okay, they might be used in their religious and spiritual purposes, okay, they might have certain powers or certain scientific applications. All right, so now what happens, these companies take advantage of this knowledge, and then they will try to own these products in the name of patents. Okay, so whenever that is occurring naturally and it has come without any proper documentation, it is the human history, it belongs to human history, or it belongs to natural history. It does not belong to one corporate commodity, one corporate companies or something like that. So these companies, what they do, they use genetic materials like genetic materials and plants and etc. And then what do? They develop for their needs, okay, which are used by farmers and indigenous people for a very long time. Understood? And then what will happen? That means they are taking advantage of this naturally occurring knowledge, okay, of human history or your biology, and then they will try to make money out of it. One of the examples we have is basmati rice. Okay, basmati rice, or use of recently it became a huge issue where turmeric was also patented as by companies thinking that they discovered the medical properties in turmeric. And turmeric has been used for thousands and thousands of years in Indian culture. Okay, and they are known for their anti-cancerous, anti-aging, and as well as antibacterial properties. Correct? No, we have very thorough knowledge about turmeric. That is why we have been using for a very long time. Neem is used for cleaning your teeth, and they called us pagans or peasants for using neem to wash our mouth and brush our teeth. Right? So neem has very exceptional antibacterial properties, and it prevents plaque formation in your teeth, and it It preserves oral hygiene. So many of your toothpaste now has neem picture on it, and nobody is telling we discovered neem's medical property. No. Similarly, we had basmati rice. Basmati rice is very famous and one of our prize jewels of our country. Why? Because it has very beautiful aroma. If somebody is cooking from very far, also we can smell it, and a lot of flavors. Flavors actually, boom out of this rice, and it makes it more special. Dish becomes more special, and it is not anybody's property. Why? Indian farmers have been developing, okay, and growing these varieties. Almost 27 varieties of basmati we grow in our country, right? So it is our crown, our jewels. It is. So now what they did, an American company in 1997, they used this basmati and they crossed with a semi dwarf variety. So a semi dwarf variety is, it will be a short plant. It will produce more basmati rice. Okay, and then what they did was they tried to patent it. Okay, using US Patent and Trademark Office. Okay, US Patent and Trademark Office. What they did, they patent this new variety. So that means what, our Indian farmers lose the rights on growing basmati. It's our own plant, and finally we lose the right of growing our own plant. And only with those companies' permission we can use that particular crop only, semi-dwarf variety of basmati. So this should not happen. So whenever it is a naturally occurring plant and you own it and you claim the novelty of this particular plant, okay, so that destroys the history, that destroys the natural, gifts that has been provided by our environment, from our nature, correct? No. So these particular things that these companies use, okay, the methods these companies use to own this knowledge and own these products and own this, history. So that has to be prevented. So this misuse of this knowledge or products or, some biologically occurring plants and animals, so that is called as biopiracy. Understood? So a lot of bodies have been established to really research about our traditional knowledge, our culture, and what has been used and how it has been used. Correct? No. And all this knowledge has been preserved. Nowadays, it's been given very primal importance. Correct? So why this problem occurs? A lot of countries, Western countries, or which are highly developed countries or industrialized nations, they have lost a lot of history and culture as they have moved forward. But when you see developing and underdeveloped countries, still the indigenous way of life is very well preserved, and this culture has been benefiting humanity even today. Correct? No. So this knowledge, this very rich knowledge becomes a platform for misuse, and that we term as biopiracy. Understood? Okay. So these things should be prevented, and unauthorized exploitation of biological resources and traditional knowledge. That is why, for example, when you take our country, Indian Parliament, they passed the second amendment. Okay, they passed a bill and they did second amendment, and this amendment was on Indian patent bill. And what do they do in this amendment is they take consideration of they include, for example, whenever a person wants a patent, they include what are the patent terms. Okay, what should be the terms that you can get the patent, and what are the emergency provisions whenever it's trying to breach a patent. And there are special teams and investigative departments that are set up to conduct research and development, okay, to understand whether this is originality of knowledge or it is stolen knowledge. Okay, so this will prevent misuse of resources. So anyways, so this is about biopiracy. So now we have a question. Exploitation of bioresources of a nation by multinational companies without authorization from the concerned country is referred as biowar, bioweapons, biopiracy, and bioethics. Okay, I'll launch a poll, students, so you can answer. Okay. Give me a quick answer. Come on, it's a very easy question. We just spoke about it. YouTube students, you can also put it in your chat box if you have not understood any topic, or if you had any difficulties. Okay, please try to communicate. It becomes better for us. All right, your time is up, students. So I will try to end the poll. Okay. So just now we spoke about the misuse of biological resources or bioresources by multinational companies to make profit, and that is called as biopiracy. Okay, remember this. And it has been asked many times, even in board examinations as well as some state examinations, state level examinations. Anyways, thank you, students. So let's end the session today here. Okay, so it was a small topic. So try to keep it natural and try to think, what could be the applications. So if you just try to use your brain and create ideas, you will understand the entire topic. Okay, so have a nice day. I'll see you in the next class tomorrow. Bye-bye.